Here are the internal structures of two cells, the antigen-presenting cell APC and the CD4 naive T cell, which will be activated by the end of this pathway. There are two main signals needed to activate the T cell. This is the first one. The APC has a structure called the major histocompatibility complex type 2, which presents the antigen ligand as a peptide. The T cell has a similar structure called the T cell receptor, TCR. Once TCR interacts with the peptide and MHC, the CD4 protein made up of 4 subunits interacts with the beta chain of the MHC molecule and assists in stabilizing the reaction. The second reaction occurs between the CD28 protein and the B7 protein. Before we talk about how these signals lead to the cellular response, here are some of the major proteins that we will be talking about. The zeta protein chains in homodimer form have 3 ITAMs on each chain. A protein homodimer is formed by 2 identical proteins. We then have one CD3 protein made from CD3 epsilon and CD3 gamma, which has 1 ATM on each. On the other side, we have another CD3 protein made from CD3 epsilon and CD3 delta, which has 1 ATM on each. The CD3 proteins are heterodimers since they are formed by 2 different proteins. Finally, we have the LAT protein, which stands for linker for activation of T cells. Due to the first signal, the LCK protein attaches to the cytoplasmic tail of the CD4 protein. Then, it phosphorylates all the ITAMs on the zeta chain and the CD3 protein next to it. This causes the ZAP70 protein to attach to the ITAMs. The ZAP70 gets phosphorylated and then it activates and phosphorylates the LAT protein. The LAT activates the SLP76 protein and it binds to it through the GADS protein. TEC protein then binds to the SLP76. From here, many different pathways are driven which lead to different cellular responses that work together to activate and proliferate this T cell. One such pathway is the MAPK/ERK pathway, which activates transcription factors that induce the expression of genes involved in T cell proliferation. The second signal is used for activating other pathways in the cell.